Good afternoon, Dr. Nandha Kumar, sir.
Good afternoon to all.
Very good afternoon. We'll continue with the next scientific session. We have with us our chairperson for today. Can I get the next slide? Dr. Manjunath, sir, Professor and Head, Department of Anatomy, Sri Aurobindo Medical College and PG Institute, Indore. We welcome you, sir, for the wonderful session. Sir has graduated his MBBS, MD, DNB, and has also completed his advanced course in medical education. Sir has right now is working in Sri Aurobindo Medical College and PG Institute, Indore, affiliated under Sri Aurobindo University. The various positions through which Sir has reached this height is taking up the Vice Dean Academics, have been acted as the head of the medical education unit, has been the medical education unit member, NMC Regional Center, AIIMS, Indore, has been NMC observer for various medical colleges, has been board of study member and paper setter for both UG and PG for various universities, and working in the Department of Cytogenetics at AIIMS University. Sir has guided PhD scholars, postgraduates, and MSc students. Sir has presented and published several articles in both reputed indexed national and international journals. Sir has also presented oral presentations in many national, state conferences and national conferences in the field of anatomy as well as in medical education. We welcome you, sir. We have our next chairperson for today, Dr. K. Radha, Professor, Department of Anatomy from BMMC, Karaikal. Madam has completed her MBBS from Perundurai Medical College, has completed her MD Anatomy from Raja Mutaya Medical College, Chidambaram, has completed her DNB, and recently has completed her PhD under VMR of Salem. Her prior experience includes she has served in Karpaga Vinayagar Institute of Medical Science, Madurandagam, as well as in Sri Venkateshwara Medical College and Hospital, Puducherry. Madam has got a total 11 years of teaching experience. Her professional achievement and awards includes she has attended many conferences and presented paper, has won best paper awards in conferences, has participated as guest speakers in several CMEs. We welcome you, ma'am.
Thank you.
Now I request the chairpersons to introduce our speaker for today. Dr. Radha, ma'am, you can introduce the speaker.
Good morning to one and all. First, I thank the management and organizers for giving me the opportunity. So I am very glad to introduce Dr. Nandha Kumar sir, because he was my best friend and we had some time working together. So I know better him. So it's very happy. I am very happy to introduce him. Nandha Kumar sir has, sorry, 1 minute. His educational qualification is he has achieved PhD in the field of medical genetics from JIPMER, Puducherry, and Master of Medical Science in Anatomy from Kasturiba Medical College, Manipal. And he has also done European Advanced Diploma in Classical and Molecular Cytogenetics in University of Montpellier in France. His awards were numerous. He has completed advanced postgraduate course in medical, that is molecular cytogenetics. He is an American board certified international cytogenetic technologist, member of editorial board for pharmacology, toxicology, and biomedical reports. And he is also a peer reviewer for electrophoresis, Journal of Psychiatric Research, and Journal of Pharmacology and Pharmacotherapeutics. He is also received senior research fellowship from the ICMR, and he presented papers in national and international conferences and given scientific talks in national level conferences. He had more than 24 publications in national and international journals. Apart from all these things, I have to add that he's a very good hard worker and he has achieved this position because of his hard work and also patience. Sir, I welcome you for this, sir. I am very eager to hear your lecture, sir.
Dr. Nandha Kumar, sir, the stage is yours.
Is my screen visible?
Yes, sir. You can continue, sir.
Good afternoon to everyone. It's a glad moment for me to be among you. And thank you, Dr. Radha Madam, for kind introduction. At the outset, I would like to thank Dr. Vishnu Bhatt, who is the research director. I happen to be one of his mentees at Japan. And according to me, he is human form of Almighty. I also thank Vice Principal, Sir Dr. Rajan. He is known for his work ethics, and he's second to none when it comes to work ethics. I also thank Professor and Head of the Department of Anatomy, Dr. Ilankathir, for giving me this opportunity to present my learning in front of you. You can, as you can see the slide, my topic is about next generation cytogenomics. Before we get into cytogenomics and the next generation aspect of the cytogenomics, I'd like to take you into the past and then talk a little bit about how we have reached to the current status. And from there on, we will take up regarding the cytogenetics aspect. So cytogenetics is study about chromosomes, and any research or diagnostic aspects involving chromosome is considered under the field of cytogenetics. And if you can see the cell and then the nucleus, you can see rod-like structures called as chromosomes, and they are visible only during a metaphase. And the chromosomal aberrations are broadly classified into constitutional, which are seen right from birth, and also acquired, which is seen in some cancers. Okay, so to give you a brief overview of what I'm going to talk today, I'll quickly go through what were the achievements and landmarks which happened in conventional and molecular cytogenetics. Then we talk about cytogenomic testing, where I will be talking in detail about NIPT, which is non-invasive prenatal screening test. And I'll also be talking about chromosomal microarray, and I'll throw some light into the new technique, which is called as optical genome mapping. Okay, so talking about brief history, we've been looking at chromosomes for the last 140 years, and out of which 74 years were spent just to identify the total number of chromosomes, which is like almost half we have spent on identifying the chromosome. Why do we think? Why do you taken so? There were few challenges. The first one is to visualize the chromosome itself, because the optics were not that strong to visualize the chromosomes. And since the chromosomes can be seen only during metaphase, we need to have dividing cells. And moreover, once the cells were obtained, chromosomes were all inside the nucleus, and then they used to clump on each other, and it made it very difficult to visualize. And scientists work on a few solutions by bringing in a mitogen, which is phytohemagglutinin, which will stimulate the cell growth. And then they used colchicine to arrest the cell culture in metaphase. And they used hypertonic solution, which is a landmark in cytogenetics, which helped to spread the chromosomes. And then later on came in staining and banding techniques. I will show you some pictures. You will understand it through the pictures which I'm going to show. So this is a chromosomal preparation, and you can see chromosome clumped on, and it's lying one on each other. Once they used hypertonic solution, from here they achieved this stage where the chromosomes spread, and then they could count the numbers. And then with this, they found out that the total number is 46. And later on, very quickly, they started banding the chromosomes. And then you could see chromosomes banded with dark and light regions in the chromosome. This appears something to be a barcodes given to chromosome, and they are all unique for each chromosome. And once they did it, they could do a karyotype, arranging the chromosome from being long to the shortest, and X and Y chromosomes arranged at the last. So once they achieved this, then they very quickly identified the basis of trisomy 21, which is 3 copies of 21st chromosome. Then they identified a Turner syndrome, where there's only 1 copy of X, which is the monosomy of X. Then they could give the cytogenetic basis of Klinefelter syndrome, which is the 2X and 1 Y chromosome. And then they could also see some gross deletions in the chromosomes, such as deletion. Here you could see the deletion of 5p arm, which is the Cray-Duchat syndrome. And also translocations between chromosomes. Here I have highlighted translocation between 22 and 9. This is a reciprocal translocation. And this is what is called as a Philadelphia chromosome. And this is the first chromosomal association which was found in cancers. Then this is a Robertsonian translocation, where 21st chromosome is seen sitting on top of 14th chromosome. If you count the number of chromosomes, it will be 46 only, but then functionally the person has 3 21st chromosome, and he'll be phenotypically, he or she will be phenotypically a Down syndrome individual. Okay, back to history. So scientists were looking at chromosomes, and then they were finding out gross changes in the chromosome. But then they were unable to look into the cryptic chromosomal changes and submicroscopic changes. Then came in DNA sequencing technology and PCR technology. Using this, what happened was the scientists were able to produce oligonucleotides which were tagged with fluorescent probes, and they were used to hybridize the chromosomes. And that was the birth of fluorescent in situ hybridization, which is a molecular cytogenetic technique. So a quick word about fluorescent in situ hybridization is where a oligonucleotide sequence of DNA is taken, and it is labeled with fluorescent probes, and then the fluorescent probes are hybridized with the chromosome, and then you can actually visualize the specific chromosomal areas in the karyotype. But for this, we need to know where we have to exactly look into, and then we have to know the DNA sequencing of that region of the chromosome. Okay, I will show you some pictures of fish, where here you could see the metaphase chromosomes. They're all blue in color. They've been stained with DAPI. But then you can also look at the green color signals, which are nothing but the centromeres of the chromosome. So what has been done here is the centromeric region, the sequence of the centromeric region has been taken, and then it has been labeled with fluorescent probe, and they've been used to highlight the centromeric region only in order to count the number of chromosomes. Then the same technique can be used to highlight the telomeres. They are called as telomeric probes. And then we can also use locus-specific probes for specific arms of the chromosome. Here, a 5p arm is being highlighted. And then came in another technique called as whole chromosome paints, where all the chromosomes can be assigned a specific color. Then once the whole chromosome painting came in, then they were able to find out the submicroscopic and cryptic cytogenetic changes. Here in this picture, which is matched with the karyotype, you can see that there is exchange between 3q region and then 10q region, which is seen here. Some part of the pink area, which denotes the 3rd chromosome, is with the chromosome over here, 10. And then from 10, you can see a part of chromosome translocated into 3rd chromosome. So these were the achievements which were made subsequently. And then for getting these kind of metaphase, they took a lot of time to cell culture. And then there came in another technique which was called as interface fish, where the culturing part was taken off from the equation, and then they were able to do the fish directly on the interface cells. So these two pictures shows this is a nucleus. Blue color structure that you see is a nucleus stained with the DAPI, and then here they have used 13th chromosome probe, which is 2 in number, and then they have used 21st chromosome probe. Here it has 3 signals, which means this denotes trisomy 21 in the specimen obtained. Usually this interface fish is used in prenatal diagnostic techniques where you need rapid results from amniocytes or from chorionic villus sampling. Okay, so then so far, what is the reality? Conventional cytogenetics gave an overview of the genome, but at a high resolution, that is 5 to 10 million base pairs. It involved tedious process of cell culture, and it required expertise eyes to find out chromosomal abnormalities. It took long turnaround time, that is about a week to 10 days, and it was not able to detect loss of heterozygosity and uniparental disomy. Then molecular cytogenetics came in with improved resolution, but it was not useful to see the entire genome. It was only locus specific. We have to know the target, then only we can design a probe, and then we can look into the target. But even molecular cytogenetics technique was not helpful in detecting loss of heterozygosity and uniparental disomy. So with this reality, the expectations that were there during those times was that they needed a technique which was genome wide, and which could have a high resolution or ultra high resolution. It should not have any cell culture techniques involved, and it should have a short turnaround time. And the sensitivity for copy number variations should be close to 100%, and it should be able to detect loss of heterozygosity, uniparental disomy, and other cryptic changes. And artificial intelligence and computer assisted data interpretation would be helpful in analyzing the whole genome. These were the expectations. So going back to the history one final time, so that was when the Human Genome Project was started, and it was completed in 2003. And that is when the genomic era was born. Then the cytogenetics techniques and molecular cytogenetic techniques, which had its own pitfalls in the form of low resolution and targeted coverage. It took up the positives of the genomics, which could do a whole genome coverage at ultra high resolution. That was when cytogenetics joined hands with the genomics, and that is the birth of cytogenomics. So cytogenetics combined with genomics is what is cytogenomics. You must have seen these books which are seen usually in the cytogenetics laboratory. This book is titled International System for Human Cytogenetic Nomenclature. This is it has 10 volumes. It has been published from late 1980s to early 1990s, and in 2013 volume it was called Human Cytogenetic Nomenclature. Whereas in 2016 volume, they have officially changed the name from cytogenetics to cytogenomics. That is to recognize the genomic input into the cytogenetic techniques. Even the latest ISCN 2020 has the terminology cytogenomics incorporated in it. And then now the cytogenetics field and then the genomic field is working in hand to hand to provide better cytogenetic genetic testing. Now we are moving into cytogenetic testing. I'm going to talk about 3 testing platforms. Number 1 is non-invasive prenatal screening. So non-invasive prenatal screening is actually done with the maternal plasma. Before I talk about it, a quick information regarding the burden of genetic disorders in India. So as per this table, we can see that around 88 lakhs to 9 lakh children are born every year with genetic disorder. So there are screening tests and diagnostic tests which can be done prenatally to diagnose genetic disorders. The screening tests are non-invasive, whereas diagnostic tests are invasive. Screening tests, they have low detection rate and they have high positive rate, whereas diagnostic tests have high accuracy and high detection rate. Now comes a test which combines the best features of both screening test and diagnostic test. It is non-invasive and it is having high accuracy. And that is what we call it as NIPT, which is non-invasive prenatal testing, where the fetal DNA can be analyzed using maternal plasma. So you can see here in the picture, this is the placenta. In the placenta, we have syncytial trophoblast, and the cells which the dead syncytial trophoblast, it releases fetal DNA, which enters into the maternal plasma. So DNA in the blood can be found in two areas, one inside the cells and the other one outside the cell in the plasma. The DNA which is seen outside the plasma is called as cell-free DNA. So in this picture, you can see a white color area highlighted, that is the fetal DNA, and then the pink color DNA shown, that belongs to the mother. So there is mixture of both fetal DNA as well as maternal DNA in the maternal plasma. So the plasma blood is the mother's blood is withdrawn, and then these cell-free DNAs, they are all sequenced. So the cell-free DNA is not a long DNA sequence like genomic DNA. It has maximum of 200 to 300 base pairs. They're all sequenced using a massive parallel sequencing technique. And then each and every bit of chromosome of the DNA which is circulating in the mother, after sequencing, it will be designated to the respective chromosome. Now, let me show you the next picture, where in normal pregnancy, if you take any chromosome, they will be having the bits of DNA in the multiple of 2. Whereas if there is an aneuploidy, then the bits of DNA which are circulating will be in the multiples of 3. So that is the principle. If you have 4 copies, then it could be a tetrasomy, and then even the monosomy can be identified using the NIPT. Okay, so now presently at the given moment, NIPT can detect only chromosomal anomalies which are related to 13, 18, 21, and 6 chromosomes. There are also some techniques which claim to detect microdeletions and copy number variations, but they are not validated. But chromosome number 13, 18, 21, and 6 chromosomes, they have a very high positive predictive value, and then the sensitivity is close to 99%. So who can avail NIPT? Anybody who has a high risk pregnancy, who are 35 years and more, the people who had a previous pregnancy with aneuploidy, positive maternal serum screening test, and any abnormality found in the ultrasonogram can go in for NIPT. But as per the ACOG guidelines, that is American College of Obstetrics and Gynecology guidance, it is recommended that this NIPT is offered to all pregnant women, regardless of their age and risk factor. But in India, the population is heterogeneous. One shoe cannot fit all. So it all depends upon the cost, and then the availability, and then the pay, and then the acceptability of the patient also. So only in India, it is only given for high risk cases. This is an NIPT report where you can see they give a report only for 21, 18, 13, and then the 6 chromosomes. So here the result comes out as whether it is high risk or low risk. As per this result, there is a high risk of having trisomy 21. And please remember that in India, this test does not report the gender of the fetus. Of course, it can detect the gender of the fetus, but it is not reported in India. The cost of doing NIPT is approximately 10,000, and the turnaround time is about 7 to 10 days. In Indian setup, only the private labs are providing NIPT. I don't have information about any government setup providing NIPT tests in India. If there are any positive findings, like in this case, there is a association for trisomy 21, and that has to be validated using invasive tests like amniocentesis or chorionic villus sampling, depending upon the gestational age. This is a very good informative chart which shows that the NIPT, okay, what is seen here in pink color is the number of amniocentesis, and green color is the number of chorionic villus sampling in a year. So the NIPT technique was introduced in 2011. And after that, we could see that the number of cases which goes in for invasive testing has drastically and significantly come down, which shows the effectiveness of NIPT. So less invasive procedure, less burden or less chance of iatrogenic injury to the developing fetus. So what NIPT cannot detect, other than aneuploidies, it cannot detect congenital malformations, spina bifida, or any other congenital, any other anomalies related to heart or kidney. And it can also go, it cannot go to single gene level. It cannot do single gene disorders. It is just screening test for only for detection of aneuploidies. So after with completion of NIPT, we move on to chromosomal microarray. So chromosomal microarray is indicated in the conditions which are all mentioned here. But basically, if you cannot come to a clinical diagnosis of any known genetic disorder, then you have to go in for chromosomal microarray. So as per the recommendation or consensus statement from American Journal of Human Genetics, chromosomal microarray should be the first tier of clinical testing for individuals who have developmental disabilities or congenital anomalies. Even for autism spectrum disorders, they are recommending chromosomal microarray rather than going in for a G-banded karyotype, because chromosomal microarray at the end of next few slides, you will understand it is a versatile test, and then it could diagnose, it could pick up what karyotyping can pick up, and even more than what conventional cytogenetic tests can do. Okay, the indications for prenatal conditions which require chromosomal microarray are increased nuclear translucency, abnormal maternal serum screening, any markers seen, soft markers seen in ultrasound scan, oligopolyhydramniosis, and previous history of genetic disorders or pregnancy. Chromosomal microarray is actually a high resolution technology. Here it can go up to 500 kilobases. Karyotyping can go up to 5 million to 10 million base pairs. Here it can go up to 500 kilo base pairs, just 5,000 kilo base pairs it can reach. And it mainly detects copy number variations. What are copy number variations? Like insertions, deletions, duplications, and amplifications, which are submicroscopic, which cannot be detected by karyotype or by FISH, can be detected by a chromosomal microarray. When the chromosomal microarray is integrated with short nucleotide polymorphisms, it can also detect aneuploidy, uniparental disomy, loss of heterozygosity, mosaicism, and maternal cell contamination, consanguinity. The list keeps going on. It is a very versatile test. Just to give you an overview of how it works, chromosomal microarray comes with a chip that you see on the right side of the slide. It is a 1.3 centimeter into 1.3 centimeter chip. If you can see here, it says Cytoscan 750K, which means it has 750,000 small wells where the DNA molecule, reference DNA molecules are already pasted. And then the sample DNA and the control DNA will be fluorescently labeled, and then they will be allowed to hybridize. If you take one section of this tile, and then if you can zoom it, you will be able to see some colors which are produced. I'll be talking about how the colors are produced in the subsequent slide, and that will be read by a fluorescent capturing device. Okay, so this is the slide which is going to talk about the basis of chromosomal microarray. So what you see here, this is the chip that I showed you in the last image. And we have both patient's DNA and then a reference DNA, which is a control DNA. Patient's DNA will be labeled in green color with fluorophore, and reference DNA will be labeled with in red color using fluorophore. So both of them will be mixed together, and then they will be allowed to hybridize on a specific area. So depending upon which has more copy numbers, they go and then hybridize. For example, if the reference genome, if it has more copy number, it goes and hybridize, and then that area will be seen red in color. Conversely, when patient DNA has more copy number, that will go and hybridize, and then that will give a green color fluorescence. So when in what all conditions patient DNA is going to have more copy number, like for example, if there is amplification or if there is any duplication, if there is copy number increase, then the patient's DNA will go and then there is a competition between patient's DNA and reference DNA. Patient's DNA will go and then bind, and then it will give green color fluorescence if there is copy number increase. If there is copy number decrease, then the reference DNA will be more in number, and that will go and bind, and it will give a red color. So if both patient copy number and the reference copy number, if they are same, that is normal. If they are same, then they both hybridize equally, and then they give a green signal. So here we can see on the right hand side of the slide, yellow color is seen when there are 2 copies. Green color is seen when there is increase in the copy number. In case of amplification, it could be aneuploidy also. And then red color is seen in the tile when there is copy number decrease, which can be seen in deletions, or it could be monosomy as well. So then these fluorescent signals are captured, and then we will be getting a graph like this, where there is an increased peak, green color peak, when there is chromosome gain or when there is genomic gain. And then when there is a genomic loss, in case of deletions, we'll be seeing a red color fluorescence coming out of the tile. So this is the principle of chromosomal microarray, and this is the workflow where DNA is extracted. See here in chromosomal microarray, no culture is involved. Extract the DNA, label it with fluorescent probe, hybridize it into the chip, and then wash it, read it with fluorescent scanner, and then there are softwares to read the report, and then you could designate whether the copy number variation is pathogenic or normal. So chromosomal microarray reporting usually is done under these parameters which are shown. The copy number variations which is detected by chromosomal microarray can be either pathogenic, or it can be benign, which is in 80% of cases, or it could be variants of unknown significance, which means it is a variation, but the scientific society doesn't know whether it is pathogenic or benign. So when the chromosomal microarray began, there were a lot of variations of unknown significance, but then slowly, that database is being built over the period of time. Now the number of unknown variants of unknown significance being detected is reducing gradually. Now it is being designated either as pathogenic or benign, or likely to be pathogenic, because a lot of data is coming out. Chromosomal microarray data analysis is again another topic. I'm not going into nuances of how the data is analyzed. I'm stopping here with the broad framework of how the reporting is done. So there are a few softwares called Carrier Studio from one company and Chas from the other maker. Basically, what is happening is when copy number variation is detected, it is first compared with normal population, and later on it is compared in a database called as Decipher, which has all the pathogenic variants already inbuilt in it. And then the database is expanding day by day, and then the number of variants of unknown significance being detected is reducing, as I told earlier. The cost of chromosomal microarray is about 6,000 to 12,000 approximately. There are some government institutes which offer it for 6,000, and private labs offer it for 12,000 to 15,000. The turnaround time for chromosomal microarray is about 3 to 4 days. The beauty of chromosomal microarray is it can give a wide range of genomic coverage. Almost it covers the entire genome. That is the functional part of the genome. You can do it within 3 to 4 days, and then with in a cost-effective manner. When you compare to NGS whole genome sequencing, of course, it sequences the entire even each and every single nucleotide, and then it gives ultra high resolution. But then the cost for that, and then the time taken for the report to come, you can't wait, especially when you're doing prenatal. you can't wait in for NGS reports to come where chromosomal microarray is handy in those situations. Now there are a few things which chromosomal microarray cannot detect. I told about positive things of chromosomal microarray, but there are few things like balanced translocation, inversion, nucleotide repeat expansion, and single point mutations cannot be detected using chromosomal microarray. And at this point, I want to tell that karyotyping is still a gold standard technique to detect balanced translocation and inversion. And also for aneuploidy, if whatever is found, it has to be any of the structural anomalies, gross structural anomalies are always confirmed with the karyotype. Now we are moving to the next technique, which is a new kid in the block called as optical genome mapping. So optical genome mapping brings in the functionality of conventional cytogenetics, molecular cytogenetics, and chromosomal microarray like a Swiss knife pattern under the same umbrella. It joins all the three techniques together, and then it gives an output. How it does, we will see. Optical genome mapping is done by a platform called as BioNano, and it has a nano channel chip. This is an ultra high resolution imaging technology, and it is not a sequencing technology. I spoke about three techniques, which is NIPT, chromosomal microarray, and optical genome mapping. Out of the three, the chromosomal microarray and optical genome mapping, they use imaging technology, and then they do not use sequencing technology. They are different from the NGS. These are high throughput imaging technologies. They use fluorophores to image. Now, what exactly happens? I know I have completed my time. I need 2 minutes to complete the workflow. Can I go ahead?
Go ahead, sir. Go ahead.
Thank you. So the first step is to isolate DNA like that of a microarray. And then here the DNA is high molecular weight, and then it is labeled with fluorophores. Here they are not labeled with green color and red color. Here they are labeled only with green color. After that, the DNA is unentangled, and then the DNA is allowed to pass through nano channels which are present inside this chip. Nano channels are small channels, and it allows only passage of one single DNA strand into it. And then when it passes through the nano channel, so you can see here a picture where the DNA molecules labeled with fluorescence. And this labeling is done based on the single nucleotide polymorphisms which are observed in the DNA. And then they are allowed to pass through the fluorescent detector. The fluorescent detector detects the molecules, DNA molecules which are flowing in the channel. And then it has an algorithm to convert it into a molecule. And then the molecules are matched against the reference genome in order to find out the structural variations and the numerical variations, even the sequence, large sequence variations in the sample which is provided to it. And then after doing the analysis, the optical genome mapping comes out with an image something like this. This image combines everything together, where you could see the outer circle, the outer wheel represents the chromosome 1 to 23, and it also has all the chromosomal bands that is seen in a conventional karyotype. So number one, it incorporates the component of karyotype. Then when you go inside, you see another wheel. This wheel is obtained, is similar to the data which is obtained by chromosomal microarray. So this shows the copy number variations. And then when you see the third wheel which is inside, that is going to show the alleles which are also detected by chromosomal microarray. This is the allele tract. This is a microarray tract, and this is the karyotyping tract. And inside, when we go, this is exclusively for the structural rearrangements which are seen in the chromosome. Here you could see that chromosome 8 has some components which goes to chromosome 1, which goes to chromosome 2, and from 8 there is a translocation to chromosome 17 and all. Okay, so the one that you see inside here, they have shown many numbers, but normal results, they show only the events happening. This is a model, and This image is called as circus plot. This is called as a circus plot. It incorporates cytogenetic component, molecular genetic component, as well as the chromosomal microarray. I have another picture to show you the various components. Cytobands are there. You have a genes track, you have a copy number track, and there is heterogeneity track, variant allele frequency track, and also gross chromosomal rearrangement. Gross as well as submicroscopic chromosomal rearrangement tracks can also be seen here. So optical genome mapping has an ultra high resolution. That is, it can go up to 500 base pairs. It can detect balanced translocation as well as inversion, which chromosomal microarray cannot do. Optical genome mapping has come out with a way to detect balanced translocation as well as inversion. There are a lot of studies which are coming out in the last 1 or 2 years, which are in favor of optical genome mapping and confirming the diagnostic potential of optical genome mapping. So it is currently not available in India, but it is popular in the Europe and in the US. It costs about $450 to $500 and then the turnaround time is about 3 to 4 days. This is an important chart which I want to show you, comparing karyotype fish microarray and optical genome mapping, also with NGS. If you see here, most of the components are ticked by optical genome mapping. What it can't do is only it can't do point mutation. Other than that, it is capable of detecting all the pitfalls in the other techniques in the earlier generation. So this is the last slide. This is a wrap up. So classical cytogenetics, even though it has a global view of the genome, its resolution is very, very low. Whereas when you come to the molecular cytogenetics, its resolution is moderate, but then we need to know which region we are going to look in. Then only molecular cytogenetic tools can be utilized. It's in targeted region, its resolution is better. When you come to cytogenomics techniques, which we spoke about, chromosomal microarray and the optical genome mapping, their resolution is high to ultra high. They give a whole genome view in a short turnaround time, and then they are also less costly compared to that of whole genome next generation sequencing. So that is the view that next generation cytogenomics can offer. So with this, I end my talk. Sorry for extending my time, and thanks to everyone for patient.
Thank you, sir. It was a wonderful session.
Sir, good afternoon, sir.
Good afternoon.
Sir, I could not join on time because of the technical glitch. I'm extremely sorry for that. But your session was very wonderful, sir, giving an insight into the NIPT and optical genome sequencing and chromosomal microarray, and you elaborately covered. And I would like to ask only one thing. You talked about point mutations which were missing. So is there any test for point mutations, sir?
So point mutations, if it is a known region, basic PCR can detect a point mutation. And sequencing technologies, they focus on point mutations, sir, because sequencing technologies, they sequence in individual base pairs. And then they have genome-wide coverage.
Okay, sir.
But the pitfall is, but the disadvantage is they take a long time, and then it is the next generation sequencing. If you go in for an exome or a genome-wide sequencing, it's going to take time. But then of course the data which it is going to give is uncomparable. But in a prenatal setting, or where testing has to, a turnaround time for the testing has to be less, and if you want high resolution in that, microarray and optical genome mapping, which is waiting in the pipeline, could be a solution to those.
Thank you very much, sir. Thank you. Wonderful session, sir. Giving an insight into optical genome mapping is really an interesting one. Thank you very much, sir.
Thank you, sir.
Sir, Dr. Nandha Kumar, sir, we have a query in the chat box. Can I, sir?
Yes, sir. Please.
Yes, sir. How much population DNA data is required for chromosomal microarray to be effective, especially in a country with a diverse and immense population like our India?
Is it typed, sir?
Yes, sir, it is typed. I will read it out again.
Is it?
How much population DNA data is required for chromosomal microarray to be effective? Especially in a country with diverse and immense population like India.
Okay, so I assume that this question is targeted towards the reference DNA which is used for chromosomal microarray, which is like labeled in the red color. That is the control DNA. I assume it that way, and then I'm giving the answer. So this reference DNA which is used is because right now we have information only from Human Genome Project, which was done throughout the globe, and then this race specific genome information, race specific whole genome information is not available. So it is the control DNA are all standardized, and then they mostly follow they are there in the mean value of all the race. And then moreover, if at all, okay, if you have a question on reference genome, if we find out any variants of unknown significance, it is we don't depend, the test doesn't depend only on the reference genome. Once we get a report, the report is matched against a database which contains information from the entire globe, which also includes Indian population. So there is if at all something is missed in a reference DNA used for the test, it will be picked up when it goes in for genome wide matching or pathogenic variant detection, when it goes to comparing it against the database which is available.
Thank you, sir. I hope it would have answered.
I hope I answered to the point.
Yes, sir. We have also one more question, sir. I'll read it out. How can we isolate fetal DNA from pregnant mother?
Okay, sir. Basically, what happens is in NIPT, they take blood from the mother. And then after taking the blood, cells are separated and the plasma is separated. In my talk, I referred that we are doing this test using cell-free DNA, which means the DNA is not present in the cell, rather it is present in the plasma. And the cell-free DNA cannot be compared with the genomic DNA, which is present in the cells. Genomic DNA will contain, it will have the entire sequence, whereas cell-free DNA will be in bits and pieces. They'll be of 200 to 300 base pairs in length. What happens is in the placenta, the the cytotrophoblast cells, when they undergo necrosis, so they release the DNA into the bloodstream, and that enters into the mother's plasma. Generally, for anyone, when we take blood, and then when you analyze the plasma, there will be some amount of DNA of that person in the plasma. That is called as cell-free DNA. That comes out from cells which are undergoing necrosis in our own body, apoptosis in our own body. That all comes into the bloodstream, and then slowly it will be taken off from the within days it will be taken off from the circulation. And even in NIPT, I want to make a point that when you take the plasma and then isolate the DNA, 90% of the DNA will be of maternal origin. Only around 10% of the DNA isolated will be of fetal origin. And then as per the NIPT, they say that minimum there should be 4% of fetal DNA in order to do this test. If somebody is obese and if they have high BMI, the amount of fetal DNA which is present in their maternal plasma will go less than 4%, maybe 2% or 2% or 1%. In those individuals, NIPT cannot be performed, and it will return as no call, because with the amount of DNA which is present, the technology will not be able to make any prediction whether there is a genetic risk or association or not. So to answer the question how to isolate the fetal DNA, the fetal DNA alone cannot be isolated. The fetal DNA will be isolated along with the maternal DNA also from the plasma using any conventional DNA isolation technique. whatever normally how you isolate DNA, like that you can isolate the DNA. But here you will not isolate it from cells, rather you will isolate from the plasma.
Sir, the last question to you, sir.
Yes.
I think participants are very much interested. Like, is there any database available for segmental duplication?
Database to know about
Available for segmental duplication.
that database is called as Decipher. It is called a Decipher. And once we get chromosomal microarray reports, then it will be matched against normal population. Then when you see that it doesn't match into the normal population, if it looks like a variant, then you take it and then put it into. Is my screen on?
Yes, sir. You can go ahead.
Okay, then I'll show you that. You go back. Okay, here you could see first, once we've identified copy number. Yeah.
We couldn't see your screen, sir.
Okay. Should I share it again?
Yes, sir. You can share it again, sir. Yes, sir. Now it is clear.
Yeah. Okay. So here you could see there are two databases which are available. The first one is database of genomic variant, and it's called as DGV. So when a chromosomal microarray report comes out, first it is matched against normal population to see whether it's variant or not. Once you see that it is a variant, it could be a pathogenic variant or a normal variant. So you take it, you take that and then analyze it once again using this Decipher software. And the Decipher software will tell whether it is a pathogenic variant or a normal variant, or it's a variant of unknown significance. I was telling you that the software is being, the information is being added on to it, and then the sensitivity of the software of the database is increasing with the amount of information which is coming inside. So these are the softwares which can be used to detect pathogenic variant, or even to say whether that variant is normal or Vivo US.
Thank you, sir. Thank you, Dr. Nandha Kumar, sir, for the brief explanation. Now it's time to honor our speaker today. Thank you, sir, Dr. Nandha Kumar, for briefing about on. Thank you, sir. Thank you.
It's a pleasure being with all of you, and then I thank all the attendees and especially all the participants for supporting basic medical sciences.
Thank you, sir. Thank you. Now it's time to honor our chairpersons today, Dr. Manjunath, sir. Thank you, sir, for being our chairperson. We would also like to extend our thanks to Dr. K. Radha. Thank you, ma'am, for being the chairperson for the scientific talk today. Thank you, ma'am. Thank you, sir. Hope everybody has enjoyed the scientific talk today. With this, we end our scientific talk for today. We have our Young Research Scholar Award, which will be starting from 1:45 p.m. And we also request everyone to join our valedictory function today. We'll be starting it around 5 p.m. today. Thank you, one and all. Thank you.
Thank you very much, sir.
Thank you.